right, let's talk a little bit about pathogen control measures. The FDA is very blunt about how to control pathogens. We try to keep them out, we try to prevent their growth, and we try to kill them. So those are the three basic ways that you're going to control pathogens or biological hazards in your food products. What about control methods for killing them? This can include things like pasteurization, not only of items that we're commonly referred to as pasteurized, such as milk, but also things like tree nuts. We can also do retort cooking, which is what we do in the case of canned foods, for example. So that's a very heavy-duty thermal process. Freezing is used to control some food products that we're not going to cook. For example, sushi. Sushi is often sourced from wild fish, and it can often contain a variety of parasites, particularly parasitic worms. So there is a freezing process that is done to render those food products safe. Pickling, or any kind of a fermentation process, is done to lower that pH and to control microbes or kill them. So pickled foods like sauerkraut or pickles are made in that fashion, and that's how they are preserved. And of course, consumer directions. We cannot forget the consumer because many of these food products, such as fresh vegetables, raw meats, are not going to receive any real kill step for pathogens. And so it is imperative that the consumers have the best information as to how to safely handle those products and make sure that they don't cause illness in the home. Preventing multiplication, again, if the organisms are not killed, we want to do everything we can to prevent them from multiplying. So I just gave an example of, for example, raw meats or poultry or fresh vegetables. Those items are typically going to be refrigerated because we don't want the microbes to grow before that product is consumed. We might also use, for example, low pH, some sort of a fermentation process or acidification process. A low water activity. So we might have something like a raw tree nut that hasn't been pasteurized, but if it's kept at a low water activity, we're not going to have multiplication of the pathogen. And any other preservatives such as salting. For example, salted meats. This is, again, one of the earliest ways in which humans preserved foods that were otherwise perishable. And of course, finally, we have our angry little friend here saying keeping them out. You want to minimize contamination in these food products. Now, be mindful of the fact that I'm not saying you're going to eliminate them. We know that these food products are often sourced from the real world, whether it's a wild-caught salmon or fresh produce that's grown in the field, and so they're going to be exposed to microbes. But we want to do our best job to minimize contamination. So when we bring raw materials into our facility, we want to make sure that we have controls in place, either supplier verifications to make sure that these practices are safe on the supplier side, possibly testing when that raw material is brought into our facility to make sure it's at its best condition and lowest level of microbial contamination. Other things that we might do include sanitation. The sanitation practices, cleaning and sanitizing in our facility, are designed to try and reduce the levels or eliminate the levels of those pathogens that might be introduced. Remember, we're bringing a raw product or a raw material into our facility. People are walking into the facility or forklifts are coming in every day. There's going to be contaminants slowly coming in, transient organisms coming into that facility. Our sanitation practices are what we use to eliminate those and to keep those numbers at the lowest possible level. And of course, package integrity. If it's a packaged food product, we want to make sure that there are no rips, tears, seam cracks, any of these other issues that could allow entry of a pathogen from the outside. Again, we want to target our controls. So again, know your enemy. And this is from the FDA's Preventive Controls Draft Guidance, Appendix 3, issued in January of 2018. And here are some examples of things that we might look at when we want to know our enemy. For example, Bacillus cereus. We know that there's a maximum temperature for its growth. And it is commonly an organism found in scenarios where perhaps the food is not kept quite warm enough. And so, for example, in food service environments, it can actually grow and cause foodborne illness. So we see that it has a maximum temperature of 131 degrees Fahrenheit or 55 C. If we keep that food product at a temperature beyond that, then we should be able to control this organism. Below it, we have Clostridium botulinum E. And it's interesting because the different strains of Clostridium botulinum have different properties. The non-proteolytic strains, which I believe botulinum E is, are not only able to grow under anaerobic conditions and are not only able to tolerate extreme heat, but they're also able to actually grow at fairly low temperatures. So if you notice, the minimum temperature for growth is surprisingly low for a Clostridium botulinum, 37.9 degrees Fahrenheit. I mean, that's refrigeration temperature. So if you have a risk for that organism being present in your food, you need to think about that control measure as well and keep that food below that temperature. In Listeria's case, again, it's able to grow at a very, very low temperature. It's good at growing in cold storage conditions, so that's something to think about if you may have Listeria contamination. The other thing we're looking at on the final part of this chart is Staph aureus toxin formation. Staph aureus is an intoxication. It causes illness through the toxin and not necessarily through the growth of the bacterium. So those toxin production temperatures are really what you want to look at. It's important to understand how we can target the controls we use in our food processing to really minimize the chances of these pathogens occurring or growing or causing illness. So if we know, for example, that we have a real possibility of Bacillus cereus causing foodborne illness in our product, we want to keep the temperature above 131 degrees Fahrenheit, which is the maximum temperature for its growth. Bacillus cereus commonly occurs in food service settings, for example, in things like cooked rice products, in which the temperature is kept below that threshold, that maximum threshold for growth, in that danger zone, and the organism is able to grow and produce a toxin. So if we keep it warm enough, if it's a hot food, we should be in good shape. Let's look at Clostridium botulinum E. This is one of the non-proteolytic strains of Clostridium botulinum E, and we know that this organism is able to grow at fairly low temperatures. This is an unusually low minimum temperature for a Clostridium botulinum strain, so 37.9 degrees Fahrenheit. If this is a cold stored product and it is anaerobic or absent oxygen, we want to keep it very, very cold in order to prevent the possibility of CBOT from growing. If we look at Listeria, for example, we know that that organism is legendary for being able to grow in refrigerated products. So we want to keep that temperature below 31.3 degrees Fahrenheit. That's that minimum temperature for growth. And if we're looking at Staph aureus, the critical factor there is the production of toxin. That's what makes people sick, rather than the bacterium itself. And so, for example, we know that there is a minimum temperature of 50 degrees Fahrenheit. So if we have a cold stored product, and we can keep it below that temperature, and we have a relatively low water activity in that food product as well, we know that we can control that organism. And if you notice, Staph aureus toxin formation occurs at a much lower water activity than some of the other examples here. So that's another factor to think about if we have the possibility or the risk of staph production. Let's look at the effect of temperature on L. monocytogenes growth. This is a very good example of the fact that as we warm organisms up, their metabolic processes speed up and the generation time, or the time it takes to produce new cells, drops dramatically. If something is cold stored, it takes a very long time, even close to that minimum growth temperature, 33 to 36 hours for one cell to produce two cells. But as we warm that product up, the time gets shorter and shorter. And by the time we get to 70 degrees Fahrenheit, it may only take an hour or two for that cell to divide and create another copy of itself. So in a relatively short period of time, we can have a product that could spoil or cause foodborne illness. Well, we've reached the end of this section, so let's go through a quick summary of what we've chatted about. Again, biological hazards may vary significantly by food and by process. You need to evaluate the risks that are specific to your operation. Know your product, know your commodity. Identify the effective control measures that might be used to minimize the growth of that organism or cause illness. So think of that Fat Tom slide that we discussed. And remember that a science-based approach to this is critical. If you don't have that level of scientific knowledge about the product, get help. There are lots of resources out there, including persons such as myself or my colleagues at Safe Food Alliance.